Welcome everyone. I'll allow folks to jump on. Just give it a moment for people to jump in. Really looking forward to today's webinar. I think it's going to be very interesting. Forgive my voice. I'm recovering from a long-term cough situation, but I'm sure I'll recover eventually. I love one of the folks who just jumped on has C9 mutant in her name. Really appreciate that. Proud C9 mutant here too. And well, just to welcome everyone, my name is Jean Swidler. I'm the Executive Director of Genetic ALS and FTD and the Legacy. We are a patient-led organization dedicated to the needs and interests of the genetic ALS and FTD community. And we are so excited to be hosting this wonderful researcher, Professor Janice Robertson from the University of Toronto. And with that, I'll let her take it away.
Yeah, thanks, Jean, for the invitation. I'm absolutely thrilled to be able to share some of our work as a slightly different viewpoint on a contributory factor to what may be going on in C9 ALS FTD. So I'll just share my screen. Okay, and there's myself over here. My Twitter handle is there. My email address is there as well. If anybody has any questions that are not asked during this session, please feel free to reach out. I'm really happy to engage with everybody in the community for me to learn and how that might help us design our research. I'm at this building here. It's called the Kremble Discovery Tower. It's in the heart of Toronto. I'm in the James Hunter and Family ALS Laboratory. Jim had SOD1 mutations, so I'm very familiar with the impact of genetic forms of ALS. I'm in close communication with his family, and they have been fantastic in supporting our research. This is our team as it stands right now. We have some new graduate students that have been added to it as well. So, yeah, I've been working on amyotrophic lateral sclerosis primarily. I was working in the UK at a place called the Institute of Psychiatry, and I trained with very well known neurologists in the ALS field, Nigel Lee, and also with Chris Shaw, who some of you may know, also in the UK. He and I trained together. And over the past several years, as we now know that ALS and frontotemporal dementia are really intrinsically connected, I have a very strong interest in frontotemporal dementia as well. And as we know, there are some genes that seem to be closely associated with a pure ALS-like phenotype disease, and those that are associated with frontotemporal dementia, clinical symptomatology, tau progranulin, and then the key gene that was found by Brian Traynor and Rose Rademacher in 2011 as C9orf72, which has really substantiated the link between ALS and frontotemporal dementia. As everybody on this call knows, there's repeat expansions in intron 1 of the C9orf72 gene, and that expansion can have a number of repeats. And we can see that on these southern blots where we have individuals with two alleles, one from mom, one from dad, that may have repeats that are within a normal range, inverted commas. We don't really know what the cutoff is for what is pathological and what isn't. And then in people who have the expansions, you can see these thousands of repeats that can be present in one of the alleles of C9orf72. And there's been, as Brian and Rose Rademakers had described in their original papers in 2011, there is proposed to be three potential mechanisms by which these repeat expansions could cause disease. There's two gain-of-function mechanisms, which is the repeat RNA-mediated toxicity, where the expansions cause these loops outside of the DNA or the RNA, and then these bind RNA binding proteins. And this can be seen here using probes that detect these repeat expansions, and it's thought that the sequestration of these RNA binding proteins is causing changes in gene expression that contribute to the disease. And then the very popular dipeptide protein toxicity generated through repeat-associated non-AUG translation that was originally discovered by Laura Ranum, and then you can generate these dipeptide proteins that can be detected with antibodies. And as you probably all are aware, there's 5 different dipeptide proteins you can see using specific antibodies, and you can see these brown aggregates that are considered to perhaps cause toxicity. And then there's the loss of function, which I'm going to be focusing on today. This is a primary part of my research program, and I'm going to give you some of the reasons why I've been focusing on this particular aspect. So DNA forms RNA, forms the protein, and for C9orf72, there are 3 mRNA species, most common. There are other ones. The types of ways that the DNA can be processed, you can get many, many different types of transcripts. They haven't all been characterized, but these are the 3 primary transcripts that are characterized, and this generates 2 proteins in humans, C9 long and C9 short. You think we'd have a better name than that, but they have the same sequence at the start, and then the C9 long has this additional sequence to the end. In other species, rodents, rats, mice, they have this protein C9 long, but they do not have C9 short. They have their own variations of the C9 protein. And it's very well established that the repeat expansions cause a loss of C9orf72. And this is caused by a transcriptional downregulation of the repeat-containing RNA. And we and others have shown that in looking at brain tissue, that there is a loss of C9 protein this is patients that we have through our clinic that carried the repeat expansions and passed away with ALS. And you can clearly see that when you have non-C9 ALS, there's the normal C9 protein, and it's lost in when you have the repeat expansion. And so I've been really lucky when I came to Toronto, established a collaboration with fantastic people, Dr. Lauren Zinman, who's the director of the ALS clinic at Sunnybrook Health Sciences Centre, Julia Keith, who's the neuropathologist who we work with through our autopsy program, and a fantastic geneticist that I work really closely with as well, Dr. Katerina Ragaeva, who has been looking at the genetics and epigenetics of many genes, including C9orf72. And we've had this collaboration ongoing for over 15 years. And through this, the Lawrence Clinic and all the fantastic people who come to his clinic and are so gracious in donating their tissues, there was one particular family that had come to his clinic where this lady had come to the clinic with ALS, no family history of disease, and so it was considered it was sporadic. And then another sister came to the clinic, and then we realized that it's not sporadic, it's familial. And then through examination, in fact, this other sister here has the expansion as well and has recently passed away as well. And then these were non-identical twins where this sister here fortunately did not have the expansion. So they lost 4 of their 5 daughters to ALS. And so, you know, we didn't know what was going on with the parents. And so when we investigated that, and as you know, mom and dad, you've got 2 alleles, and then you have the normal allele, and then you have the repeat expansion allele. And then this can be seen here, is that when we look at the parent, one of the parents, there was 70 repeats. And so it was actually the father carried 70 repeats. And from the 70 repeats into the next generation, it went from 70 to hundreds of repeats. And what was really interesting was that although all the daughters had passed away with ALS, the father passed away at 90. and with no ALS, no frontotemporal dementia, died of natural causes. And this was really interesting to us and to our understanding of how these repeats are contributing to disease. So, however, when he passed away, and this family has been terrific in providing tissues, they provided to us also internal organs. We were able to look at the repeat expansion in all different kinds of tissues. So although the father had 70 repeats in his blood, when we looked at different regions of the brain, so here's the normal allele here, here's the 70 repeats, and you can see that it starts to kind of disappear. That's because it's expanded in different regions of the brain. So it's got different lengths in different regions of the brain. And this is called somatic mosaicism. So he had 70 repeats in his blood, but he had repeat expansions in his brain, but he did not have disease. So then we wanted to look at the gain of function mechanisms and loss of function mechanisms in these tissues. And so we, as I say, we were very fortunate to obtain tissues from this family. And when we looked at dipeptide repeat proteins, this is the father and this is the daughter who passed away with ALS. And I think you can see quite clearly there are tons of dipeptide repeat proteins in this particular region of the brain. This is in fact the cerebellum, but when we looked in other regions of the brain, there were equivalent numbers of dipeptide repeat proteins across the different types of dipeptide repeat proteins in the father who did not have ALS or FTD, who died at 90, compared to the daughter who died in their late 50s. So they had equal numbers of dipeptide repeat proteins. They had equivalent numbers of RNA foci, but what was really surprising was that the father had increased levels of C9orf72, not decreased levels of C9orf72. So for some reason, the 70 repeats led to an increase in C9orf72, but it had dipeptide repeat proteins and RNA foci. So the major difference that was happening here was that there was increased levels of C9orf72 in the father. And so we want to understand what C9orf72 is doing, because if it does have neuroprotective properties, or if you don't have it and you lose it, we need to know exactly what it's doing so we know how to develop therapies. And so this is the amino acid sequence of C9orf72, and what we would do is take this sequence and put it through various programs, and nowadays it's the amount of technologies that are available to analyze these sequences is just fantastic. And so using this, you're able to work out domain structures that have been characterized in other types of proteins. And what became apparent from C9orf72, it had these different domains within the protein structure. And these are DEN, UDEN, CDEN, and DDEN. And DEN is a horrible name. It's differentially expressed in normal neoplastic cells. And C9orf72 contains these domains, and the C9 short isoform also has one of these domains. And C9orf72 is now classified as a tripartite DEN domain protein. So what are DEN domain proteins? Well, DEN domain proteins are usually involved in membrane trafficking. And I just want to show this little video of, these are all different little organelles and vesicles, and this is all going on in all of our cells right now as we're sitting here. All this movement is going on inside of our cells. And as you can see that, you know, when these membrane vesicles hit like the outer membrane, they fuse, and then it releases other things into the in-between cells, and it helps in targeting proteins for degradation. And so C9orf72, as a DEN domain protein, is involved in trafficking of these organelles and vesicles within a cell. The issue is that we don't exactly know which membranes C9orf72 is regulating, because if we don't have C9orf72, we would expect there's going to be problems in this membrane trafficking. So a big question is, which membranes is C9orf72 regulating? So a few years back, we made antibodies, specific antibodies that we could detect C9orf72 in tissues, human tissues. And the first thing that we observed was that when we label tissues, human tissues, with C9orf72, and here we have a neuron here, and the brown staining is where we see C9orf72, you can clearly see here it's circling the nucleus, here too. And then we use fluorescence labeling. This blue dye here labels the nucleus, and the green here is the C9orf72 labeling the nuclear membrane, as it's called. And so what we found by comparing healthy tissue, people who did not, I don't like using the word normal, but it's control, or people who did not die of ALS, when we labeled with, this is a motor neuron labeled with C9orf72, you can see the nuclear membrane is nicely labeled, but in someone who passed away with the repeat expansion, you can see that that nuclear membrane labeling is lost. And so therefore we considered that if you don't have C9orf72 at the nuclear membrane, what is it doing there? Is it causing disruption of the nuclear membrane? And so this is, just want to really show you, this is a cell, this is the nucleus. And within the nucleus, there are these little pores, and this is how things are trafficked in and out of the nucleus, proteins, RNA, and this is what keeps, this is the machine of the body, of the cell. This is where all your DNA is, this is where your RNA is made, and then it's exported out of these pores, and proteins are exported out, and other things are exported in. So you want to have good functioning of these nuclear pores. And they're complicated structures. There's lots of proteins that are involved, but this is the basic machinery that's called nucleocytoplasmic transport. And so you have a cargo protein, for example, that binds to these proteins called importins. They are then delivered into the nucleus, where they then encounter this protein called RAN-GTP, and this causes a dissociation of the protein from important beta, and it keeps the protein in the nucleus, and then the important is re-trafficked back out into the cytoplasm again for another cycle of nucleocytoplasmic transport. And so what we had found is in previous work that C9orf72 interacts with both importin beta and with RAN-GTP, two fundamental proteins involved in the nucleocytoplasmic transport machinery. And so the question was, what's happening to importin beta and RAN-GTP in these scenarios where you've lost C9orf72? And so what we found was these are motor neurons. And so we've done a lot of our work in C9orf72 knockout mice. And I know that there's a lot of work using iPSC human motor neurons, which are really valuable. But all models are important. And the reason that we've been using also the mouse model is because you have a system. It's the full system. You've got all the other cell types there. It's all organized in the way that it's supposed to be. And you can learn a lot about the biology of these proteins in that animal model, even if it does not necessarily, it's not a human, it's not recapitulating everything, but every model has its pluses and minuses. And so we feel that we can learn a lot about the biology of C9orf72 in the animal model. And so this is a motor neuron in the spinal cord of a mouse, and it's labeled with important beta. And I hope you can see here that when you do not have C9orf72, you get all these granules here. So the nuclear membrane is here, and then important beta shouldn't be here in these levels. But if you don't have C9orf72, then it's causing a disruption of important beta, which is a protein that's important for nucleocytoplasmic transport. The other protein that we looked at was RAN-GTPase. This is a driver of nucleocytoplasmic transport. Here you have a normal motor neuron, or a motor neuron from a wild-type animal, and a motor neuron from an animal that does not have C9orf72. And you can see that there's more red in the cytoplasm here. That is more of this RAN-GTPase in the cytoplasm, and it shouldn't be there. And then finally, we actually measured nucleocytoplasmic transport. And this just shows here it's a gradation. There's more green here in the nucleus, and what this is using a nucleocytoplasmic reporter, that there's deficits in nucleocytoplasmic transport caused by the loss of C9orf72. So C9, this is one of the membranes that C9orf72 seems to be involved in, and its loss is causing disruptions in the nuclear membrane. And so one of the questions for a lot of any illness, or if you even think about people with COVID, people who get long COVID, people who respond, who have vulnerabilities to different things, that you might have a genetic makeup. And I think we're learning a lot from long COVID in terms of the immune system, how it deals with these things. And some people, you know, have increased susceptibility to having negative responses. They don't recover as quickly. And so I think this can apply to a lot of things. And so here we have the house on the rock, and then the house in the sand. Everything's well and good until the tide comes in, and then this house will fall apart. And so what we were wanted to investigate is whether the problems with the nucleocytoplasmic transport in the C9orf72 knockout mice, would they be more vulnerable to some kind of insult? And so one of the things that we did, we injured the neurons in the C9orf72 knockout animals. And this is TDP43, a key protein in ALS and FTD. And what we found was that when these neurons are injured, you get these TDP43 granules in the cytoplasm when you don't have C9orf72. And after the injury, the wild type animals, they recover normally. So it could be that, you know, if you think about some kind of traumatic injury, that if you don't have a proper functioning nuclear membrane, then if you have injury, then you're going to get more TDP43 in your cytoplasm. And in a normal scenario, that would return to the nucleus as the neuron recovers. But there's a possibility that if you have a problem with your nuclear transport system, that TDP persists in the cytoplasm and could cause problems. And so we're continuing to pursue this avenue of research. So I just want to change to a different membrane now that we're incredibly interested in, and that is the synaptic membranes. And so neurons communicate with each other through synapses. And so here we have one neuron here sending signals down, and it needs to communicate with another neuron so that signals like in movement or thought, those signals can be delivered. And this occurs through these processes here. They're called synapses. You have the presynaptic terminal. So this is the presynaptic neuron. And then you have the receptor neuron, the postsynaptic. And glutamate, the signaling happens through a process called neurotransmission, where you have neurotransmitter release from the presynaptic neuron. So it's glutamate comes out, it's released from the presynaptic neuron. And then that glutamate interacts with glutamate receptors on the postsynaptic neuron. And then there's a whole cascade of signaling events that then transmits that signal depending on what function is required. And one of the major glutamate receptors that we've been focusing on is a glutamate receptor called AMPA receptor. And they are composed of different types of subunits. And one of the subunits is GLUR1. And so it's known from neuroimaging studies, from transmagnetic stimulation, that there are problems in neural communication that occur in C9 repeat expansion carriers before the onset of disease. And work out of UCSF had shown that they can measure in repeat expansion carriers that there's something going on. Their estimates are that it can occur 10 to 20 years before the symptom onset. And, you know, this is true of Alzheimer's disease, Parkinson's disease, SOD1 mutation carriers as well, that there's something going on pre-symptomatically, and these are really important points for intervention. This would give this is crucial information. And if we can understand what's going on at those points, then we could put in therapies that would even prevent the symptomatology. And so we had, using our antibodies, we found that C9orf72 is localized to synapses, suggesting that it is involved in the communication between neurons. So here we have a motor neuron, here I've outlined it here, and all these little dots that I'm showing you here that we verified, and we published this work about 4 years ago, is that C9orf72 is present within the synapse. So what's it doing there? And so we looked at what happens in mice again, because they're a very tractable system. If you don't have C9orf72, what we found was that there's a change in the makeup of these AMPA receptors. You end up getting more of this GLUR1 subunit, and it's quite clear. If you look in the wild type situation, there's the GLUR1. You have more when you have half a copy of C9, and when you have a complete knockout of C9orf72, you have more GLUR1. And work from Justin Ishida's lab and from other labs have shown that in iPSC neurons, here you have iPSC motor neurons. You can see there's GLUR1 levels in a control motor neuron. And then when you have knockout of C9orf72, you're getting elevated levels of the GLUR1 on the surface. And this is also true in the full expansion carriers. So these are knockout motor neurons. These are patient-derived from repeat expansion carriers. And these phenotypes, or this elevated C9 GLUR1 on the surface, can be rescued solely by increasing the levels of C9orf72. So losing C9orf72 leads to this. And also, if you replace C9orf72, it rescues it. And studies from looking at synapses in human motor cortex, again, C9 expansion carriers have elevated levels of GLUR1. And work again from Justin's lab had shown that these cultures that have this elevated levels of GLUR1 on their surface, they're more sensitive to glutamate excitotoxicity, which has been, for the ALS field, been a theory, a hypothesis that's been going on for a number of years, over 20, maybe 30 years, but it hasn't been really nailed down exactly what is going on. And so what we find is, just in summary, is that in the normal situation or in a wild type situation, you have these AMPA receptors at this level. But if you have a loss of C9orf72, a reduced amount or loss of it completely, you have AMPA receptors that contain more of this subunit called GLUR1. And so cortical hyperexcitability in humans, the C9 repeat expansion carriers, is detected before disease onset. Interestingly, epileptic seizures occur at a higher rate in people with FTD than the general population. And it has been reported in asymptomatic C9orf72 mutation carriers. This may reflect the underlying cortical excitability caused by abnormal glutamate receptor function observed in pre-symptomatic ALS FTD. So is it possible that when you have lowered C9orf72 before disease occurs, that it's acting through elevating the number of these glutamate receptors, and these elevated numbers of glutamate receptors make it that there's more sensitivity to a second hit like excitability. And so we asked the question, are the C9 knockout mice more susceptible to epileptic seizures? I mean, if this is the case, is this going to happen in C9 knockout mice as well? And so we induced seizures in knockout mice. We induced it with canic acid, which is a very well-established agonist of glutamate receptors, including AMPA receptors. And we measured EEG. This is just the electrical activity. And I think you can see really clearly that the knockout have far more electrical activity in their neurons compared to the wild type. And we measured this here as well. So the C9 knockout mice, when they're dosed, they'll have, it's assessed over about 60 minutes. And we can make these recordings and we can see that these animals have, not only do they have higher seizure activity, but the seizure activity lasts for longer. So the important thing here is that we dose these animals at 3 months of age. So then we waited. So we got 1 dose of canic acid at 3 months of age. And so we waited till the animals were 10 to 11 months of age. So then we analyzed these animals. And so at 3 months of age, there isn't much difference in the weight. But what we began to see is the animals got older, that the C9 knockout animals began to lose weight. These are animals that were treated with canic acid. They start to drop off in weight. And then we did a whole battery of behavioral tests. So we did rotor rod to assess motor function. This was not significant, but you can see it's trending down. We did a grip strength analysis, which is significant. With the C9 knockout mice treated with canic acid, you can see that they have, that's a measurement of motor performance. They have reduced mobility compared to wild type animals. They have memory deficits. So this test is really significant, is that a mouse always wants to explore a new object. So it starts off, has the same object that it will explore, and then you replace one of the objects, and then you measure the amount of time. The wild type animals treated with canic acid, the C9 knockout animals treated with canic acid, how long do they spend looking at each object? And the C9 knockout animal doesn't remember that it had seen object A before, so it looks at them at equal amounts of time, whereas the wild type animal spent much more time exploring the new object. And then the other test that we've done is that there's less memory of an incident. So we have this noise, and the animals will freeze upon hearing this. They get a shock, and we measure the amount of time the animals freeze. And the C9 knockout mice have less freezing time. This is called cued fear testing, to see if they remember, if they hear a noise, that they will get a little shock. And the C9 knockout mice have less ability to remember that. So 1 dose of canic acid at 3 months in knockout mice versus wild type, and the knockout mice go on to develop motor and cognitive phenotypes that in the animal modeling world are modeling ALS FTD. So this brings us again to this 2-hit hypothesis, is that these mice, they live, you can't tell the difference in these mice in any measure. Wild type mice, C9 knockout mice, if you look at them, and they seem completely normal until you challenge them with something like canic acid, and then you wait, and then the C9 knockout mice start to develop all these phenotypes that have been activated by excitotoxicity. So the loss of C9orf72 predisposes to this second hit. And so what we want to know is, what is the mechanism by which C9orf72 is regulating these GLUR levels? Why is there more GLUR1 here when you lose C9orf72? And so we've been doing this. We moved from looking at the whole animal, looking at neurons in culture. And what we found was that if you compare wild type, heterozygous knockout, and full knockout, that when you don't have C9orf72 there's reduced dendritic outgrowth. And we can measure this by a process called show analysis. You measure the length and the number of intersections of all of these processes that are coming from the neuron. And they are reduced in the knockout compared to wild type. The heterozygous is reduced compared to wild type, but the effect is exacerbated when you have complete loss of C9orf72. And the other thing that we had found in the animal model, and that had been found in iPSC neurons, is that when you have loss of C9orf72, you have elevated GLUR1 levels as well, which you can see here, and this is quantified here. And then importantly as well, when we do a glutamate excitotoxicity, that's second hit. When we do that, and we can look at live cells using calcium AM, which is green, and the dead cells are BOBO3 iodide is red. You can see in the control situation there you have your neurons all look nice and healthy, and then you add glutamate, and you can see all the red here, and there's red here as well, that these neurons, the knockout, C9orf72 knockout neurons, are more sensitive to glutamate excitotoxicity. They're more sensitive to that second hit. And so then what we're trying to figure out, okay, so you lose C9orf72, you get more GLUR1. So what is going on in terms of how these AMPA receptors are trafficked to that surface? And there's a lot of, it's very, very complex process. And but one of the pathways that we've become very interested in is glutamate receptors are trafficked by the actin cytoskeleton, which includes a protein called cofilin. And worked in the Huntington's field, this paper was just published, I think, last year, is exactly the kind of pathway that we're going down. So these lines and all this branching here, this is actin. It's the actin cytoskeleton. It's involved and it's regulated by this limb kinase cofilin pathway. And if you have a disruption of this pathway, you can change the number of AMPA receptors that there are on the surface. And that can influence not only the size of these structures, which are called dendritic spines, but also their responsiveness to glutamate-induced excitotoxicity. And so when we looked in our C9 knockout animals and in our knockout cultures, you can see that if you look at, and the particular cofilin that we look at is that this limb kinase here phosphorylates cofilin. So this is kind of an abnormal event that occurs. And if you look in the heterozygous and the knockout animals, you can see that there's an elevation of phosphorylated cofilin when you don't have C9orf72 there. And that coincides with elevated GLUA1. So there you have the GLUR1 or GLUA1 that's used interchangeably. That's elevated when you don't have C9orf72. And that elevation coincides with the elevation of phosphorylation of cofilin. So this is a process that we're really been interested in. It kind of provides a mechanistic explanation as to why there's changes in AMPA receptors and why there's changes in GLUA1, and that is causing this increased vulnerability to glutamate excitotoxicity. And so this slide here says that small molecule targeting cofilin reverses the deficits caused by loss of C9orf72. So we've been working with a small molecule that targets this phosphorylation of cofilin. And we're quite excited by our results that if we treat our C9 knockout cultures with this small molecule, we reverse the deficits in neuritic outgrowth. This is all the quantification. The one that I really want you to look at is the pink here is the knockout. And then the black line, which is up here, is the C9orf72 treated. And even in the wild type treated, you can see that there's also an improvement. It's very good for the neurons. It reverses the elevated GLUR1 levels. So here's a C9 knockout. It's greener here. And if you treat them, you can see that these look a bit dimmer. And we've measured this as well, that the treatment targeting cofilin normalizes the levels of GLUR1. And importantly as well, as I showed you that if you treat C9 knockout neurons with glutamate, it kills most of the neurons. But if you have the treatment targeting cofilin that rescues the glutamate, the GLUA1 levels, then you rescue the effects of glutamate excitotoxicity. And so I don't want to go into lots of detail in every single aspect of the project. I'm really keen on questions and anything that you want to ask me, but I just want to give you a review of what I've talked about. Is that the C9orf repeat expansion causes reduced C9orf72 protein. we reported a family in which the parent with 70 repeats had abundant DPRs and RNA foci and passed away at 90 with no ALS or FTD. The major difference was that the parent had increased, not decreased, he'd increased C9orf72 levels. C9orf72 appears to be involved in membrane trafficking. We're trying to figure out what membranes it's involved in. It seems to have a fundamental process and different pathways. And we've established a role in nucleocytoplasmic transport and neurotransmission. And I don't think that these events are not exclusive. I think they are dependent on one another. And we're in the process of investigating that. So loss of C9orf72 causes elevated GLUA1 levels and increases the vulnerability to glutamate excitotoxicity. And we can rescue that by using a small molecule that targets an actin-binding protein, cofilin. And we are proceeding with that work in iPSC neurons and organoids and hoping to develop that for not only the preclinical testing, but taking it further into the clinic. So some thoughts on what I've talked about. I really, our work is really suggesting that that there is this second hit, and if you think about even sports injuries, you know, certain individuals, even with chronic traumatic encephalopathy, that some individuals succumb to that. Some people can get one concussion and recover, and another person can get a concussion and are very ill for a really long time. And there's a lot that we have to learn about that in terms of neuronal functioning. So I think what our work is suggesting is that there are ways that we could bolster the house. Let's get this house up on the rock, and let's do that as soon as we can. And one of the potential ways of doing that might be to elevate C9orf72 itself as a therapeutic strategy. So I'm going to end there, and I'd be happy to take any questions. Oh, and I'll just finish with my acknowledgements. Sorry about that. I'm really grateful to our funding bodies. I'm really grateful to Jim Hunter and his family. This is our core research team, and there's two young PhD students that are just been phenomenal in this work. One is Bilai, who has done all of the canic acid work in vivo, all the electrophysiology, and a new graduate student who just joined just over a year ago and has just been phenomenal in terms of providing the data and having a good understanding of glutamate receptors and next steps. Thank you.
Wow. Well, thank you so much for the super fascinating talk, Professor. And there's some great questions in the chat that I'll read out to you. But from the position of privilege as the facilitator for the event, I wanted to ask if you had thoughts about whether, you know, there's a few states for people who are in this position, and just this father and the family that you've, you know, done so much work on. There's people with the mutation who are fully healthy, and then there's people who are preclinical, who the disease is developing with them, but they appear healthy. And there's people with disease. Do you think it's possible that the father was in the preclinical stage and just died naturally before the symptoms appeared, or do you think that he was fully healthy?
Well, he did not have any, we examined his brain completely. So there was no signs, no, completely healthy. So one of the things, when we talk about this a lot, if we all live long enough, we'll get cancer, right? And so that's just an inevitability. And so if we live long enough, will we all get Alzheimer's disease? I don't know. But, you know, if he had lived longer, I mean, that's the open question, you know, but all I can say is that, I cannot answer that question.
Yeah. Just
love to answer that question.
Would love just love to hear.
It just shows you the importance of these types of studies and the graciousness of these families who've helped us to do this. And we need more families like this so that we can really investigate. And when you said preclinical What did you mean by
Oh,
preclinical
biomarker active, but no symptoms,
biomarker meaning?
like TDP-43 issues, NFL rising, other things like that.
Okay.
There's some great questions in chat, so I'll just start reading them from top to bottom. From Teresa, it seems that the goal in the C9 trials has been to selectively knock out the expanded allele. What are the most significant differences between wild type and the heterozygous people with the knockout, or mice with the knockout, or whatever?
So they're not knocking out the expanded allele. The antisense oligonucleotides were to suppress the expression of the repeat expansions. So the antisense trial that was run by Biogen, if that's the question, was to knock down DPR and gain of function mechanisms. That's what that was. They haven't cut out the repeat expansion. They were just knocking out, they wanted to suppress the production of the dipeptide repeat proteins, which they did, which was successful. They did knock down the dipeptide repeat proteins. So the difference between the heterozygous and the complete knockout in the mouse models is that I think it will take longer for the C9 heterozygous mice to become susceptible to glutamate excitotoxicity, etc. But we found that when we look at the heterozygous in a culture system, they're responding. They have, although they have, it's a gene dosage effect in terms of the levels of GLUR1, for example, and the responsiveness to glutamate excitotoxicity. So I think it's a dose effect. But the other thing as well is that we don't know what cell types you lose C9orf72. It could be that neurons don't have any, and the rest of the C9 is somewhere else. So we don't know where we're losing that C9orf72 protein. And the dipeptide repeat proteins are primarily in neurons. They're not in other cell types. And so there's something about the neurons. They're not dividing, they're not correcting it. So it's building up in there. So I hope that helps answer your question.
Wonderful. And Candy asked, how do you lose C9orf72?
That's a great question. We had published that as well. I didn't want to so what happens is that the repeat expansion, so we have something called epigenetic modification. So you get methylation of certain nucleotides, and CPGs, these islands are within the repeat expansions, and they're also on either side of the repeat expansions. So if that becomes methylated, then it shuts down the expression of the protein. So what was really interesting was in the father with the 70 repeats, his repeat expansions were not methylated. So his C9orf72 could go on expressing unimpeded.
Wonderful. Could you share how your small molecule is delivered?
So the small molecule, that was done in culture, that was added to the culture. So if we're doing it in vivo, it's actually, you can take it just by IP in vivo.
And for those of us who don't know what IP is.
Oh, sorry, just to yeah, sorry about that. Yeah.
Intravenous.
Yes, intravenous. Yes. Sorry.
Okay.
Yes.
No, thank you. I learned something new, another thing new.
Yeah.
So from Brandon, is there any understanding of what causes the transcriptional downregulation of C9? I've read some research discussing hypermethylation of the promoter region, but why does this happen?
Because it does, it's a control. So it happens in Huntington's. It happens in all of these repeat expansion diseases. Somehow your system recognizes that as that repeat expansion is not good. And so the cell mechanism sees it, and that methylation process happens to shut down maybe the expression of the dipeptide repeat proteins. But at the same time, you can lose expression of the normal protein. So it's a response. And also we have methylation, you have changes in your gene expression. So you can have twins, and we had published that as well, is that you have twins and they get the disease at different ages. And that can also be due to lifestyle choices. smoking, drinking, whatever. And this is also something called biological aging. So you can have your chronological age and your biological age, and your biological age can influence an advancement of the onset of disease. So that work is done by my colleague Katerina Rogaeva, doing fantastic work in that realm.
Wonderful. How long before you believe your small molecule will make it into a human trial?
That's a great question. We have a lot of preclinical work to do. I'm hoping sooner rather than later. It's actually already used for other purposes, so we just need to get the preclinical data in place before we're talking with our colleagues on Monday actually about that. How can we accelerate this to get this into the clinic?
Fabulous. Good luck. Any idea if conclusions can be drawn from specific repeat expansion numbers and their relation to symptom onset?
That's a great question, and that is the burning question. How do you work out what the lengths of the repeat expansions are, in your brain, and how do you assess that? And what cell types have them? Can you have more than one length of repeat when your RNA is generated? Do you have multiple repeats within the same cell? So if we're talking about what length of repeat is necessary for disease, we do not know the answer to that question. All we know is that the repeats are multiple lengths.
Can I tell you, Professor, something I saw at the ALS MND symposium? There was some new long read sequencing from PacBio.
Yes.
In the poster, they showed a C9orf72 carrier over time that his repeats changed over time in the blood. And I thought that was very fascinating. And I don't know if you have any thoughts on that, whether this mosaicism might be happening not just in the brain, but maybe elsewhere too.
Yeah, I think that's interesting. And what happens in your blood does not tell you what's going on in the brain for sure, but the repeat instability is pretty well known in PacBio. I know that there's other groups who I think are looking at repeat length in a single cell approach from brain tissue. So even in a cell, even in the iPSC cells, that it's expanding, it's expanding, contracting, you've got DNA repair mechanisms going on. So it's really variable.
As someone, Kenny asked, as someone who has C9 repeat and diagnosed with the ALS, and this is more of a general question, Professor, what are the chances of FTD developing? And does that make progression faster, if you're aware of that? I know that that's not a clinical is not really your field, but.
Kenny, nobody knows the answer to that question. I think, Jean, you would agree with that. Nobody knows why one individual gets ALS, why another individual gets FTD. It's really not clear why that happens.
I think people diagnosed with both do have worse progression, maybe, but they were already diagnosed with both when those observations are made.
ALS, I think the reason that I think ALS has a faster disease progression than the FTD component, as far as I'm aware, depending on the type of ALS. Yeah.
And Brandon has another question. Do you have any thoughts on the viability of the? I can't pronounce this, but you can see it, perhaps, Professor. The PIC5 inhibition from Justin Achita as a way of competing for the loss of C9.
Yeah, there's other groups also using that PIC5, but he's now suggesting a different mechanism for the PIC5 that it's involved in.
I think they've said for a while that they think that it would compensate for the loss of function. I don't understand exactly how. And originally they were going to do, already one of them did it in just C9, and the other one was going to do, there's two companies developing an approach. But the other company moved on to general ALS already. But
yeah, I have open minds about it. I'll be interested to see the results of that. I can't say if because the mechanism in the original paper was that it was involved in glutamate trafficking. And then in the more recent paper, it was involved in the release of misfolded proteins. So it seems to be a complex thing. And maybe C9, this is the thing, if C9 is involved in a lot of membrane stuff, it could impact in a lot of ways. But anyway, I like Justin's work very much.
If loss of function is the cause, how come we don't hear about other loss of function mutation causing ALS? I guess in contrast to SOD1, which is clearly gain of function, or FUS with that clinical trial looks like it's gain of function. So your thoughts?
I think that's a really great question, but I think it also speaks to the possibility of a second hit. I mean, it's interesting that there was a paper that just came out in the UK that only 33%, or not only, but it's 33% of repeat expansion carriers. Oh
yeah, we had this researcher present. It's on our YouTube. It's very interesting.
Is that Amon?
No, it was people in Utrecht.
And
they didn't really account for FTD at all. They only looked at ALS. But what's very fascinating is in their paper, they had these families with very high, tons of people, huge pedigrees. They only looked at families where there was 10 kids. So they had plenty of people to look at. But there were these families with, tons of ALS and these large families. And then they had other families where there was just one person in these huge families. So it's 10 or 20 kids. And I was so curious, how come nobody's done more of the work like you did, where they've tracked down the parents to really investigate what happened there? Maybe something new happened there in the one case, the solo case. And that would really change those penetrance estimates. And even in that paper, they do accept that it's probably family specific. That these highly penetrant families that many of us have probably have something else going on.
Yeah. And also, I'm not saying that loss of function is I'm not saying that the DPRs and the other things are not important. I just think that it's important to consider all aspects. And I think maybe the loss of function aspect is understudied. And I think bolstering C9orf72 function could be helpful.
Cheers to that.
Yeah.
Somebody asked, about the UCSF work that says loss of neural function 20 years before symptom onset.
Yeah. That's Bill Seeley's work. So you'll know about all of that. They've done all this imaging. When I say neural function, it's the neural connectivity. And so what is neural function? So they're able to detect changes many years, but that's true of most of these diseases. I mean, Alzheimer's disease is now at least 25, 30 years before the clinical onset of disease. So I think understanding what is going on, and then there's also the possibility that's been raised is that everybody's got things you have to compensate for, and maybe during neurodevelopment, there's also ways in which the brain has been dealing with this to compensate for these changes.
Yeah, it's so fascinating. And for any of our viewers, we do have a guide to C9orf72 for patients, for people impacted, and we link to some of the papers that talk about what changes have been seen or documented, 10, 20 years before symptom onset. And there's a lot of work out there on that. From Candy, how do you determine the changes in C9orf72? Do you do repeat genetic testing, or is it a blood test?
How do we identify repeat expansion carriers? Is that?
I think it might be changes in the length. I would assume that this is saying.
Yeah, that's done by Southern blot analysis. Yeah.
And not very common.
No.
And then someone shared that there are SOD1 variants with loss of function, which I'm sure you're aware of.
Totally aware of, yeah.
Well, we're at the hour.
And then also, if the SOD1 aggregates, right? If you think about the mutant SOD1, I mean, it's also being considered that because the protein aggregates, then it can't work. So is that a loss of function? You know, so it's very complex about exactly what's going on. So yeah, these are fantastic questions. Thank you so much.
Well, thank you so much for coming and presenting here. I want to respect your time.
Thank
We
you,
said an
Jean.
hour. It's an hour. Any last words?
No, I really appreciate having this opportunity to talk about our work. And I'm really looking forward to connecting with anybody who wants to get in touch and talk to you, Jean, about all other things associated with C9orf72 and the impact. Thank you. All right. Thank you. Bye.